The investigation we chose to base our presentation on was looking at ways to inhibit the reverse transcriptase enzyme found in the HIV virus. This was done by adding cation compounds to the enzyme substrate. These bind to the substrate, which changes its chemical properties enough that while it can still bind to the enzyme, the reaction can't take place. This means that when it interacts with the enzyme, it remains bound to the active site of the reverse transcriptase, preventing the enzyme from interacting with the unedited substrate. This is an example of competitive inhibition.
Enzymes are biological catalysts. Catalysts lower the activation energy for reactions. The lower the activation energy for a reaction, the faster the rate. Thus, enzymes speed up reactions by lowering activation energy. Many enzymes change shape when substrates bind. This is termed induced fit, meaning that the precise orientation of the enzyme required for a catalytic activity can be induced by the binding of the substrate. Inhibitors. Enzyme inhibitors are molecules that interact in some way with the enzyme to prevent it from working in the normal manner. There are different types of inhibitors, including non-specific, irreversible, reversible, competitive, and uncompetitive. Poisons and drugs are examples of enzyme inhibitors. There are two main classes of enzyme inhibitors, reversible and irreversible, that are differentiated by the magnitude of their affinity for enzyme. Reversible enzyme inhibitors bind and dissociate with the enzyme in an equilibrium process. Irreversible inhibitors bind tightly to an enzyme to form an essential permanent complex. Because irreversible inhibitors form such a tight complex with the enzyme that they permanently remove its catalytic activity, irreversible inhibitors are also termed inactivators. Some irreversible inhibitors even become covalently bound to amino acids in the active site of the enzyme as they are brought through the early chemical steps of catalysis. Reversible inhibitors can be classified as competitive, uncompetitive, or non-competitive inhibitors. If the detailed mechanism of inhibition is known, then the classification can be made by identifying where on the enzyme the inhibitor binds, or the order with which it binds, relative to substrate. Alternatively, a determination of simple kinetic parameters can generally be used to classify the inhibitor.
Before we go into depth about HIV inhibition, we must first familiarize ourselves with the different types of inhibitors. Competitive inhibitors are inhibitors which have a similar structure to the substrate to which the enzymes bind to. Thus, they compete with the substrate molecules to bind to the active sites, causing the reaction rate to decrease. But the binding to the enzyme's active site is reversible and can be overcome by high substrate concentration. So, the Km increases but has no effect on the Vmax. The graph shows this. Uncompetitive inhibitors are those which usually bind to an enzyme, usually forming a covalent bond, which reactivates the amino acid residue near or at the active site, which causes a change in the overall three-dimensional structure of the enzyme. Thus it becomes inactive. This inhibition occurs mainly in multi-substrate reactions, where the inhibitor is competitive with respects to one substrate but uncompetitive with respects to another. Non-competitive inhibitors are inhibitors which reversibly bind to a site other than the active site, which cause a change in the overall three-dimensional structure of the enzyme that causes a decrease in the rate of reaction. However, the effects of a non-competitive inhibitor cannot be overcome. So they decrease the Vmax but have no effect on the Km. HIV stands for human immunodeficiency virus. HIV is a retrovirus that leads to the failure of a human's immune system. It is passed through sex, blood, and also breast milk. HIV causes AIDS, which is acquired immunodeficiency syndrome. There is no known cure for HIV, mainly because of its high mutation rate.
The graph shows how much of the different cation compounds remain bound to the reverse transcriptase over time. From this graph, it can be seen that the zinc chloride remains bound to the enzyme much longer than the other two, and so inhibits the enzyme's activity for much longer. The inhibition caused by zinc chloride would be the most likely candidate for further research, as it causes the longest lasting inhibition of HIV's reverse transcriptase, but it also caused the least ill effects to cells when it was introduced to them.